Welcome students to another video lecture. Today we are going to start the new series of the lectures from the Lippincott, and the chapter today is the chapter number 3, and it belongs to globular protein. As we all know that globular proteins, their name suggests that they are a globe shaped, that is globular, and they are spherical in nature. They are wide in nature, so that's why they are called as globular proteins. Now, students have a quick overview about what are the globular proteins. They are very diverse in nature. They have different structures and different functions, and that's why they perform different specialized functions. In this chapter, we will examine the structure and the function of the globular proteins and what clinically importance they are according to their structure and their function. Students, the globular heme proteins, they are called as heme, as we already discussed globular, spherical, and heme mean the iron containing prosthetic group which are tightly attached, bound to the prosthetic group. Heme means the iron containing group. These are the globular proteins which are three-dimensional structure. We have already studied in our previous lectures that the cytochrome function, it is also a heme protein iron containing, and it's present in the electron transport chain in the inner membrane of the mitochondria, where it plays oxidation and reduction reactions. It is also a heme, the globular hemes, such as cytochromes. The another one, the heme group example is the enzyme catalase, which is present in peroxisome, the organelle present in the cell, especially the liver cell, and the heme group is the part of the active site of that enzyme. Here it plays a catalytic role. Here it plays the oxidation reduction role, and here it is the catalytic role. And of course, the hemoglobin and myoglobin both are the respiratory pigments. They also reversibly bind with the oxygen, and not only transport but also some of them function as to storage of the oxygen. So, students, there are many types of globular heme proteins in our body. So, first of all, we will study the structure of the heme group. Heme is a complex of protoporphyrin 9 and iron. So first, let us discuss what are protoporphyrin 9. 9, there are protoporphyrin, according to classification, is from 1 to 9, and it is represented by the Roman numerics. Why it is called as 9, or we can say what is the identification of 9. 9 and 1, 2, 3, 4 are different from their side chains. In our case, the porphyrin 9, we must have four methyl groups. As you can see, methyl group number 1, methyl group number 2, methyl group number 3, and the methyl group number 4. Two vinyl group CHCH2 double bond. Here, vinyl group number 1, a vinyl group number 2, and two propionic acid CH2CH2COOH. Here there are two propionic acid, and these are the side chain. On the other side, the central atom is the iron, and these are the pyrrole ring number 1, pyrrole ring number 2, pyrrole number 3, and number 4. And the classification according to the porphyrin 9, these are the basic structure present in the all the heme group. But the side chains are different, so that's why the side chains having the 4 methyl group, 2 vinyl group, 2 propionic acid are the protoporphyrin 9, and the central atom is the iron. Here is the structure of the three-dimensional structure of the heme group. As you can see that the iron is present in the center and having the four bonds, and it is bonded to the four pyrrole rings. Pyrrole ring number 1, pyrrole number 2, pyrrole ring number 3, and the pyrrole ring number 4. Now the other two valencies or the bonds which are present, which are perpendicular, you can say, to the pyrrole, four pyrrole rings, that they are perpendicular, 5 and 6 mentioned in the green color. There is a histidine. The histidine is directly attached to the iron, and on the other hand, the sixth one is directly attached to the oxygen. Then we can say two additional bonds on one side of the planar porphyrin ring. These position is coordinated to histidine residue. This is the histidine residue, where other position is available to bind oxygen. So students, as we can see, there are four rings attached to the four pyrrole rings, and one is attached to the histidine, and other is for binding of oxygen. They are present in the heart and the skeletal muscle. They can act as not only as the carrier of oxygen but also the reservoir of oxygen. Myoglobin as well having the only single polypeptide chain, so that's why it attains the maximum level of tertiary level. It belongs to the tertiary level of protein because it has only one polypeptide chain. Similar to the tetrameric hemoglobin. In hemoglobin, there are four polypeptide chains, but in hemoglobin, the myoglobin, there is only one polypeptide chain. The arrangement make myoglobin a useful model to attain some of the complex properties. Here are the structure, three-dimensional structure. Myoglobin has 8 stretches from A to H. Number one, as you can see here, the A, this is the A, and there is a bending, then there is a B stretch, then here again there is a bending and C stretch, there's a D again bending and D stretch, E stretch, F stretch, G stretch, and the H stretch. There are total A to H stretches which are present into the myoglobin polypeptide chain. And why these chains are present, why these bends are present, why these stretches are present, you can see that this is the A stretch and here is the bend. The bend is due to a number of reasons. First of all, in the bend, proline can be present. Why? Because proline has the amide group and it is a side chain, and that's of the amide group because it has a ring nitrogenous group in that. So that's why it cannot be accommodated in the helical structure and it is forced to fold like that one. And it can be for beta chain as we studied in the previous chapter and the loops. So in the bends, yeah, in the stretches, proline, beta chains or loops can be present. But these bands can be stabilized by the hydrogen bonds and the ionic bonds. Here you can also see the bends. So bands are because of prolines, amino acid residue, beta bands or the loops. Now students, this is the globular structure or the tertiary level of the myoglobin. As we have previously studied in our previous courses, that whenever the tertiary protein folded in an aqueous environment, the hydrophilic amino acids are towards the water, and the hydrophobic, they are buried inside, and it is one of the contributing factor uh, that it becomes the globe, it achieves the globular shape, as with and other types of bonds are also present as disulfide bonds and ionic bonds and hydrogen bonds. But this is the location of the polar and the non. But you simply said that hydrophobic amino acids are buried inside and the hydrophilic is towards the aqueous solution or water. This is the surface lined with the non-polar, where is the location. As we all know that this is the polypeptide chain and it is spherical in nature, and there is a gap, and this gap is known as the crevice. And here the hemoglobin, our heme group, is present in this cavity. Now students, uh, come towards the bounding of the oxygen with different type of the molecules. First, there is a proximal histidine. Proximal histidine binds directly with the iron atom. This is also called as F8. Proximal histidine is known as F8 because it is present in the F stretch of the polypeptide chain, and its sequence in the F chain is the eighth amino acid. One other one is the distal histidine. It is present in the E stretch, and the number in the E stretch is the seventh amino acid, and its function is to stabilize the oxygen. The myoglobin creates a special environment for the heme, permits a reversible binding of one oxygen molecule. Now we can see that how these proximal and the distal histidine helps in the binding of the oxygen to the heme group. Students, you can see this is the this is the iron which is present inside the this is the oxygen. This is the iron, and this is sorry the iron. This is iron, and iron is attached with the four groups, and this is the iron, and iron is attached to the four pyrrole rings, and this is the histidine, which is known as proximal histidine. Now, the proximal histidine is the part of the polypeptide chain, and this polypeptide chain, and come there is a histidine, and the histidine is directly attached to the iron group. So that's why it is known as the proximal and directly attached to the, because it is present in the polypeptide chain, and because of the folding there is another histidine which will be present here. This will be called as a distal. This is the distal histidine. The distal histidine, and this is the distal histidine, and in residue in the polypeptide chain, and now we make it a globular one. This distal histidine, this stabilizes the oxygen with the iron. So we can say the proximal histidine is directly attached in the polypeptide chain, and the distal histidine is present also in the polypeptide chain, but it directly attached to the oxygen, and oxygen attached to the iron. Similarly, here you can also see that the red color mentioned here, it is the iron, and this is the oxygen, and oxygen is attached to the distal histidine, which is the part of the polypeptide chain. And similarly, this polypeptide chain, when folded again and again, comes together, and uh, this is the proximal histidine shown in the diagram, which is directly attached to the iron. So the proximal histidine directly attached to the these attachment to the iron, and this attachment to the oxygen helps to stabilize the globular structure of the myoglobin and makes the affinity stronger than that of the hemoglobin. Thank you, student. That's all for today. For any question and queries, please uh, inquire it into the comment section. We hope we will get together in the future, inshallah. Thank you.